This is a production of Cornell University.
Thank you, everyone, for coming and for listening. Like he said, I'm going to be talking about genetic load in cassava, finding deleterious mutations, and looking at what they do. As many of you may know, cassava is a clonally propagated crop through propagated through stem cuttings. And if you also know, if you've read or watched a science fiction novel or film, cloning has some costs. And one of those is embedded in the evolutionary principle. Sorry, this is not moving. There we go. Of Muller's Ratchet. Muller's Ratchet states that if you don't have sexual recombination, you're going to have an accumulation of deleterious mutations. And here, this is seen in kind of a pictorial way. Under sexual reproduction, even like disregarding anything like recombination, you can purge these deleterious mutations, here those red symbols, just through independent assortment. But under clonal reproduction, the deleterious mutation, especially if it's recessive, is masked and never exposed as a homozygote. So its effects linger, and that's the genetic load. And so one of the things we want to ask is, well, if we expect a lot of this genetic load in this clonally propagated crop, how can we find it? And so one of the ways we can detect deleterious mutations is through evolutionary conservation and comparative genomics. Well, the hypothesis being here that over millions of years, we expect that natural selection will have purged out deleterious mutations. And that if we look across species, then we should be able to see which sites are conserved. Sorry, this is a red. There might be another mic. Hello? Oh, there we go. Help. Okay, can you hear me? All right. Where was I? Okay, so if we take let's just this example sequencing cassava, and right now we know nothing about it. It's assuming everything's neutral. But if we start aligning some species, some distant diverged species, we can start to get a sense of maybe the conservation state of some of these bases. And as we get more and more species, we can even start to say, well, this red site is conserved everywhere, but we have a derived allele cassava. So we're going to predict that this is probably deleterious. And maybe there's some semi-conserved other sites out there we can't say as much about, but and then some purely neutral sites. And so one of the things also to highlight is that using these interspecific comparisons allows us to eliminate the effects of linkage or drift that we'd have by looking at stuff like allele frequencies within a single species. Also, maybe the question comes up, well, why don't you just align every species that exists and see what happens if you're trying to use evolution? Well, the problem is if we align Arabidopsis and maize and all of that to this, nothing's going to align. It's going to be very gappy. It's a little too diverged. And so what we want to do is maximize our evolutionary sampling while maintaining, a comparative biology and similar alignable space. So we did this by sampling around 30 genomes across public repositories and botanic gardens. We sampled these, sequenced them using short and long read technologies, and assembled them. And I'm kind of glazed over that, but that was kind of a lot of work on the project. But we come out of this with a big set of assembled genomes. Highlighted in red is cassava. But there's some closer and farther related to cassava. But kind of to reiterate here is we're trying to sample as much as we can within a similar evolutionary background to get both a broad, more alignable space and deeper evaluation at every base pair. And so, what do we do once we have this? Is we use a tool called Genome Evolutionary Rate Profiling, or GERP. And what GERP is, it's very simply, it's a method of evaluating evolutionary constraint. It produces something called a rejected substitution score. Basically, it assumes, or it estimates the amount of neutral mutation that would happen in a region, and then looks at a site and say, well, how many mutations should have occurred? And if they didn't occur, that's a rejected substitution, and that indicates conservation. So we take a multiple sequence alignment, put it through the GERP pipeline, and come out with this big set of GERP scores across. And the benefit here is this: as we put all these species in here, we can evaluate a lot more sites and a lot greater depth, meaning we can have more rejected substitution kind of differentiation. So what we're really interested in is these most highly conserved sites, and we expect mutations of these sites to be deleterious. And then, the question is, if we expect these are, effective, causative, functional elements of the genome, how can we use those in something like genomic prediction to improve our estimates or use that functional information? And but I do have to state some assumptions first. If using genomic prediction, especially if we're talking about the GBLUP kind of standard method, if the markers are tightly linked to causative loci, then adding causative SNPs is unlikely to benefit our prediction. So, but that isn't always the case, and we would assume that it might be beneficial to include this functional information in situations where the LD structure is broken down, but things aren't as tightly linked. And two of those situations might be in a rapid cycling recurrent selection or in a diverse or cross-population prediction. And so the method we're going to use here is called a multi-kernel GBLUP. Basically, simply put, this allows us to separate our estimated effects from the two different sets of markers, our genome-wide SNPs and then our putative deleterious SNPs. And the assumption going in here is that these are coming from different variant effects. And so these kind of assumed distributions is just kind of an example of what we think might be going on. And so then we tested in a cross-population prediction model using, we found around 10,000 segregating sites using our conserved GERP sites. And we tested this in two separate breeding programs in East and West Africa. And so we used information from IITA, a breeding program in Nigeria, to both do it within and cross-population prediction to another breeding program in Uganda at NACRI. And here's just the first results, which I'll point to you. Basically, adding the deleterious here, all these 10,000 SNPs didn't change any prediction accuracy. But we do see the predicted drop from going from within to cross-population in the different germplasm. Well, there's a lot of explanations that I'll get to throughout the presentation, but one of the big ones here is that these 10,000 markers is just too large a set, and it's likely just tracking the same variation as the genome-wide set would. And so it's not really, we really need to know is, well, what information will we, how much information will we need, at what grade could we get some benefit? And that led us to look at some simulations. So when would this be beneficial? And so this simulation framework, I basically just took the real genotypes from these populations, looked at those conserved, deleterious sites, and then simulated marker effects at those sites. I used a few different genetic architectures, basically just assuming different orders of magnitude QTL. Then from that, I simulated the phenotypes and held, kept track of what the highest effect variance would be, these highest effect QTL, and performed a genomic prediction. So the baseline is going to be using just the whole SNPs. Then I'm going to use all of the causative SNPs or these deleterious markers. Then we're going to look at just paring down to the highest effect, what happens. So here's the results. These are just the cross population. Let me walk along the x-axis for you. So here is when we don't add anything but the genome-wide SNPs. And then as we move, include all of them, and then as we go down to just the top deleterious SNPs. And the different colors are those genetic architectures. And right now we're just looking at the cross-population results. What we do see is this blue line shows that under a highly polygenic trait, even knowing that the most causative QTL, the highest effect is not going to help anything. But that's thousands of QTL, very small, and I don't expect that to be true. But we do see a big bump in the other genetic architectures, but only after we kind of narrowed in on the top SNPs, the green and red, only including the top 100, top 10, including everything does not help us. So I look at the within population as well, and I expected this to be different from the cross, but it looks very similar. So maybe my LD patterns aren't quite as different as I thought they might be, or this is just one iteration of a simulation. I'm trying to perform more to get a better grade of what this should look like. So that kind of leads us to say, well, 10,000 once again is just too big a set, and how can we get a better grade scale of what's the highest impact? So some recent work from a former lab member, Guillaume Ramstein, suggests that by incorporating protein information, we can improve the weighting of these variants at the conserved sites. And he saw significant improvements by doing this weighting and doing prediction with those high effect markers. I'm not going to go too into this, but using some information from databases like SIFT and UNIREF that track evolution and protein folding, get a random forest model that predicts conservation with a few other covariates. But this gives us these conservation weights, which is what I'm trying to get at, is a more graded scale of what's the highest impact. I'm going to just show this. This is my compare of my observed conservation versus predicted. And the only thing to highlight here is that there's an enrichment for rare alleles in this predicted versus the observed, meaning we're capturing more rare alleles are expected to be deleterious as well. So, we're hopefully getting more signal there. And fortunately, when I go to my real predictions, including these weights, has not yet given the desired boost in accuracy like I see in the simulation. And there's very many possible explanations. It could be that maybe yield looks like that blue line. Maybe it is really highly polygenic. I still don't believe that. Oh, but it could be maybe the deleterious estimates I still need to work on getting that pipeline down and tweaking. It could be that root yields in a clonal crop is not the same as grain yield in an outcrossing crop, and it may not be related to as much to fitness as that might be. Or it could be an LD still there, or there's just G by E could be this big drop. Oh. And so, there's a couple possible problems, but there's a lot of avenues of still going through, so it's not like I'm discouraged quite yet. And to include, we've assembled these genomes and we have deep assessment conservation across a huge portion of the cassava genome, including these functional information. I think it has the potential to improve genomic prediction, but there's some caveats and only under certain circumstances. But the really important is that we need to get at what are the highest effect variants. We see some difficulties, but I think there's a lot of room to improve, and we're getting there. And I'd like to thank lab members, especially from the Robbins Lab, Kelly, Chris, and Francisco for their support, and from the Buckle Lab, Ed, Cinta, Sarah, Guillaume, Amy, and Tweed. And I also thank my funding and the Next Gen Cassava community for all the work they do. And I will take any questions. From Martin, did you consider directional dominance model allowing a non-zero mean for deleterious kernel effects distribution? Might be appropriate here. I do have a dominance model. I've not looked at directional dominance. So let's talk. Joey Lee, do you think it would be helpful to include genomes outside of the Forbiaceae family to cut down 10,000 steps to find more conserved to improve your prediction? Yeah, I think it does have the potential. There are other publicly available genomes out there that are closer than Arabidopsis, but still, that could have improved the depth. It's still hard because it's hard to get a real graded score out of GERP. It's really just, this is really conserved or it's not. Even though, and there's a variation in the scores, but it has a lot more to do with how many species can align at any given site. Thank you again. If there's any questions. I probably went over time since a lot of interruptions. So,
well, let's thank Evan.
This has been a production of Cornell University, on the web at cornell.edu.